My research is to study the expression of RADIALIS RAD in Solanaceae. According to former research, RAD encodes a single MIP transcript factor implying controlling of flower symmetry in antirrhinum magus. Common name is snapdragon. Antirrhinum magus is a species of a flowering plant belonging to order Lemmios. Lemmios has a close relationship with order Solis. 4K genes has been shown to control dorsal-ventral symmetry in antirrhinum magus. Cyclodia, Dittoma, Radialis, and Deverocata. RAD is activated by the sac and DAC in the dorsal region of developing flower. It plays a major role in establishing flower symmetry. RAD encodes a 93 amino acid protein containing a single MYB-like domain. At a morphological level, RAD and DIV antagonistic. RAD promoters dorsal identity and DIV promoters ventral identity. Given the sequence similarities between the RAD and DIV proteins, the antagonism most likely affects molecular competition. Modern for interaction between genes controlling dorsal-ventral asymmetry, expression of SAC and DAC in the dorsal region lead to activation of RAD, which in turn inhibits DIV activities in both dorsal and lateral region. After knowing how the SAC and RAD expression in the laminas, I need to know how the SAC and RAD expression in the Solanaceae. According to the former study from the Jimbo, who is the postdoctoral works in the same laboratory with me, SAD2-like genes in the Cystisus pentas and Solanum lycopodcas shows the radially broad pattern during the earlier stage of the flower organ initially, and also was detected in the functional terms of the late stage of the organ mutation. I also found another research about to study FSM1, which is a close homologous to the radialis genes. These genes reduce the organ size and control the cell's expansions intervals and reduce the left arrow but not compound left shape. All these studies shows that the expressions of the SAC and RAD in the Solanaceae is different in the antirrhinum magus. All these studies may guide me to think what will RAD expression in the Solanaceae and if it also play a role of the flower asymmetry. The family of flower plants and the Solanaceae consists about 98 genera and 2007 species include tomato, potato, eggplant, and tobacco, with a great diversity of habits, morphology, and ecology. My hypothesis is expression of the RAD genes was detected in the functional terms of the late stage of the organ mutation, because I know the RAD genes is the downstream genes of the SAC and DAC in the antirrhinum magus. I hope it also be a downstream genes in Solanaceae, so I want to it has the same function with the satellite 2 genes as studied from the Jimbo. My works now is get the CDS, is the region code of the radialis from a gene bank, and I aligned them and decide the primers. After that, I will plasmate the contribution and do the genetic library screen, and finally do the PCR to analyze the expressions.